pleasure to be able to open this symposium with a talk on the use of molecular microbiology for sustainable water quality management in aquaculture. So let me just start with this image from space of the city of Bangkok. Oh, you have this, brilliant. So this is Bangkok from space, and what you can see is just the vast expanse of the coastal belt where all of the land, virtually 100%, has been converted into aquaculture farms. I'm sure you're pretty familiar with the story how Thailand, in response to the increasing global demand for aquaculture products, increased its production capacity with these high-density, low-biodiversity monoculture aquaculture ponds until we reached a kind of tipping point in 2010-2012, where we experienced something like an ecosystem collapse, and we had novel diseases that spread amongst the aquaculture ponds and which caused a reduction of almost 50% in the output from these production systems. So I think this is an example for what we would call an unsustainable food production system. And you mentioned the Sustainable Development Goals. So one of the Sustainable Development Goals is to design more sustainable food production systems. So systems that are less prone to this kind of problems. So in this context, together with KMUTT in Thailand, we applied for funding to build a link between Newcastle University and KMUTT to build capacity for the use of next generation sequencing technology for better microbial management and disease prevention in aquaculture. Now, what do I mean by portable technology? So this photo here illustrates the laboratory which we kind of assembled in Newcastle with small pieces of equipment that you can readily carry with you in order to, so we can take this equipment, we can put it in a suitcase, we can go somewhere else in the world, we can do fancy microbiology there. And the center of this laboratory is the MinION, which is a memory stick-sized device for sequencing. So how does this technology work? Basically, it allows us to read genomic information, so we can pull a strand of either DNA or RNA for one of these nanopores. As the strand passes through the nanopore, there's a change in the signal output, which allows us to read, to detect the sequence of the nucleotides. And this allows us to gain information about the kind of bacteria which we have in water samples. And the nice thing about the MinION is, you can see it on this photo, it's very small, and therefore we can use it outside of the conventional laboratory. This is a technology which we were the first to really develop for the use in water quality monitoring. If you're interested in this, I really recommend that you read this paper where we compare how well this technique performs in comparison with other ways of measuring water quality. Now, building on this work that we've done in Newcastle University, the aim of the network, the link with KMUTT, is to see whether we can apply this technology to the monitoring of microbial communities in aquaculture and make this technology accessible to aquaculture applications. So we have three objectives. The first objective was just to train people here in Thailand in how to use the MinION. The second objective was to work together so so we could co-develop the application of this to microbial community monitoring in aquaculture farms. And the third objective was to demonstrate that this can be beneficial to aquaculture producers. So in terms of the first objective of the training, the way we envision this is that we have some skills and knowledge in Newcastle. The first step is to transfer these skills to a team here in Thailand, KMUTT. But the idea is that you then become the trainers for other researchers in Thailand, but also across Asia. And eventually, these researchers, they can interact with the practitioners. So the last step is the knowledge transfer from the universities into industry. And this is just one example of how we work with international partners. So we've got people from India here where we did similar trainings, we do similar training in Africa, and hopefully we can just multiply our efforts. At each stage there's a multiplication factor, and in this way we hope to achieve a much bigger impact than we could achieve on our own. So how did we go about this? In January, we came to KMUTT, I gave a lecture, we did the first sampling together. That was in January of this year. In March, the team of researchers from KMUTT came to Newcastle and worked with us in our laboratory. And my research technician, Adrian Blackburn, provided hands-on training in the use of this green algae. And then in April, we came again to Thailand, and took samples in Thailand, and we did all of the sample processing in your laboratories. So by this point, we were able to demonstrate that this works in Thailand. And from April onwards, really, all of the work was done by the researchers here at KMUTT. And so they took the lead, they took the samples, they did all of the analysis. So I think we really achieved the first step in transferring the knowledge. We'll see the evidence for this today from Newcastle to our partner here in Thailand. Now let me just give you one example of why these modern technologies are so powerful in investigating water quality. And the example is one that has already been introduced in the introductory or in the welcome address, which is this topic of antibiotic-resistant bacteria. So if you read the scientific literature, you will see a lot of papers where people say, because aquaculture producers use antibiotics, we have resistant bacteria in the environment. So these scientists are blaming the aquaculture producers for causing antibiotic resistance. Now the farmers that we work with, we interviewed them, and they all tell us, no, we are not using antibiotics. So there seems to be a contradiction here. Who is right? The only way to find out is to go and do measurements. Then you have the evidence. So I will now show you the kind of evidence we can collect with these technologies that allows us to decide which version of events is the correct one. So these are our case study farms, and I'll show you data from the first sampling events where we just sampled the 5 case study sites on the left-hand side of this image. We would go to the sites and we would take water samples from the pond, and we would take water samples from the canal that provides water into the pond. We also take sediment samples from the pond and sediment samples from the canal. And we do it for 5 different sites, and they include extensive aquaculture, semi-intensive aquaculture, intensive aquaculture, and organic aquaculture. So we should be able to see whether the management practice has an influence on the microbial communities in these farms. Now this image, by the way, I cannot show in the UK because they think this is a safe practice here, but maybe here in Thailand, there we go. So we apply 3 different types of technologies. This is the traditional one. I'm sure you're familiar with this. This is just throwing the bacteria on the plate. This is the one I just introduced. This is sequencing. So this we can do with the memory stick size device, the MinION. And this is an alternative technology which we call quantitative PCR. So this is also looking at the genomic material that we recover from the environment. And each of these methods has some advantages and some disadvantages. So we use all of them in combination, we get much better information than if we only use one type of technology. So this is what we did for the aquaculture ponds. We look for different types of bacteria by culturing, and then we look at all of the bacteria by using the sequencing technology, and then we look at different types of marker genes, such as for Vibrio cholerae, for example, but also for antibiotic resistance, and this we do with qPCR. So these are some of the data. So this is the data from the sequencing, this is the data from the culturing, and if it's a C, it's the data from the canal, and if it's P, it's from the pond. So we have 5 canals, we have 5 ponds, and this is the number of fecal bacteria that we determined by the plate count. This is the sequencing data. So just some of the sequencing data. So what we do with sequencing, we identify 100,000 bacteria in each sample. Then we can see how many bacteria out of 100,000, for example, come from the human gut. So here we show you the bacteria that we believe come from the gut of humans or animals in different types of water samples. So if the number here is 0.003, it means that 3 bacteria out of 1,000 bacteria in this water sample are from the human or the animal gut. And finally, we've got the qPCR data, which is antibiotic resistance genes. And this is high throughput qPCR. So we can look for 250 genes that relate to antibiotic resistance in a single analysis. Now what's really striking here, if you look at these patterns, is how each method gives you the same pattern. So each method tells us that there's something unusual about these 2 canal samples. There's a lot of fecal bacteria by the plate count method. There's a lot of fecal bacteria by the sequencing method. And what's interesting, they also have a high level of antibiotic resistance genes. So we find antibiotic resistance genes in the canal and not in the aquaculture ponds. And we see a very close correlation between antibiotic resistance genes and fecal pollution indicators in the canal. So let's look at this in a little bit more detail, just to see that we can, of course, look at this in much more detail as well. So this is multi-drug resistance. Now maybe this sounds very hazardous, multi-drug, but actually these are non-specific resistance mechanisms. So this is just a response that the bacteria develop in response to stress, but it's not specific to antibiotic resistance. So it's not a good marker for exposure to antibiotic chemicals, and you can see there's no clear pattern for multi-drug resistance. But if you look at specific resistance, for instance to sulfonamide, there's a very, very clear pattern here. So it's high in the canal, in these 2 canal locations, and it's low in the ponds. There's virtually no sulfonamide resistance in the ponds. Also, it's high in the water and it's low in the sediment. So the source for this antibiotic resistance, based on this evidence, very clearly is the water in the canal. And we can see similar trends for tetracycline resistance, that the canals are the sources for antibiotic resistance to tetracycline in this environment. So that's the overall picture. High resistance in these two canal sample locations, and they are from the same canal. I'm not sure whether I spelled it correctly here. Canal, the correct interpretation. Now this canal is actually draining the city of Bangkok. And that's the canal where we find a lot of antibiotic resistance genes, and very low in the sediment. So this is the main source for resistance that we've discovered in our survey. I can show this data in a different way, which is just as a correlation, and we see that there's this strong correlation between resistance on this axis and fecal pollution indicator on the other axis. With next generation sequencing, we can also look at other types of bacteria. And this is shown in this plot here, for example, nitrifying bacteria. There's no correlation between nitrifying bacteria and resistance, for example. So the resistance in the water is linked with the fecal bacteria in the water. So in conclusion then, we have very strong evidence that the reason for antibiotic resistance in our case study area is because there's fecal pollution in this canal water, and this fecal pollution is coming from Bangkok. We see no evidence whatsoever that the aquaculture is a source of antibiotic resistance at this case study site. And to add further evidence, this is the exact same finding that we had in the preliminary study, where we looked at 3 case study sites, 2 of them which were the same ones, but we only looked at 1 resistance gene, tetracycline. The exact same conclusion that the pollution comes from Bangkok and gets into the aquaculture pond. It's not coming from the aquaculture pond into the environment. So here's my take-home message. You need to treat your wastewater if you don't want antibiotic-resistant bacteria in your aquaculture ponds. Now we all go to the toilet, every single one of us. We need to take responsibility for the pollution that we are producing. And if you don't do this, this pollution will cause problems in the environment. It will also cause problems with our food supply. Okay, so that's just a snippet, an illustration of how these technologies can provide really powerful insights that you wouldn't get with traditional methods. And we hope to be able for you people in Thailand to be using these technologies after the training and at the end of this project, which unfortunately is coming to an end. So this is the last moment. So I just want to very briefly talk about what we call project exit strategy. So this project is finishing, but hopefully the research and the impact is not over yet. So what we achieved is a lot. Within just 1 year, actually within just 4 months, we were able to teach you how to do it. And you were able to use it independently. So I think this is really a huge achievement of this link that we have between the 2 universities. In terms of transferring the knowledge to other researchers, I think we only started. There will be a workshop tomorrow, but it will just be theory. So there's still a lot to do in terms of trying to pass on the knowledge that the team at KMUTT has to other teams across Thailand and other teams across South Asia. And then the real challenge is passing it from academia to the practitioners. We had a really good workshop yesterday with the farmers, but I think it was just making them aware that, look, we've got these technologies, and it's difficult for them to understand the technology. But at least they know it's there. But there's a lot of work that still needs to be done for us to convince them that this is something that is useful to them. So I think what will be really important to me also to provide evidence to the funding council is to look at this again in, say, 5 years, in 6 years. And hopefully it will look different from where we are today. And hopefully we will have evidence that you are still using this technology, that other people that you have trained will be using it, and that the practitioners will also be using it. So that's my message going forward. Let's try and take these skills, let's keep using them, let's tell others about it, let's pass it on to others, and let's try and engage with the practitioners and show them what we can do, but help them use these technologies to solve their problems. That's it for me. I don't know how I did on time.
Thank you for your presentation.